subscribe now and press the bell icon. Never miss an update.
Good morning, students. So in the last class, the pollination had completed. Today I am going to explain about the pollen-pistil interaction. Pollen-pistil interaction. Already know about the structure of the pollen grains and the pollination. What is interaction between here the pollen and pistil? Pollen and pistil. In pollination, the pollination does not guarantee the transfer of pollen grains. The transfer of right type of the pollen grains. No guarantee. The transfer of right type of the pollen grains. What is meant by here the right type of the pollen grains? What is meant by here wrong type of the pollen grains? See here the right type of the pollen grains that is the compactability. The compactability pollen grains. Wrong type pollen grains means incompatibility. The pollination does not guarantee the transfer of the right type of the pollen. What is meant by right type of the pollen? Compactability. The pollen grains belongs to the same species. What is the incompatibility? The pollen grains belongs another species. See here, this is the stigmatic lobe. It is a stigmatic lobe. Stigma. It is another crop plant stigma. For example, these are the two crops plants. Example S1, S2. The genes of S1, S2 is the genes of the, for example, tomato. Next one S3, S4. These are the genes of the potato. Here the two crop plants are there. One is tomato, another one is the potato. The genes of tomato S1, S2. The genes of the potato S3 and S4. See here in this two stigmatic lobe, the pistil has ability to recognize either the pollen grains or it is the right type or wrong type of the pollen grain. Right type or wrong type of the pollen grain. See here in this stigmatic lobe, this is a pollen grain S1 and S2. The pollen grains is containing the genes S1 and S2. Here this is another pollen grain, for example S3 and S4. So these are the pollen grains. So this pollen grains belongs to the tomato. This pollen grains belongs to the potato. Now the both pollen grains are landing on the stigmatic lobe. So when the both are landing on stigmatic lobe, The pistil has ability to recognize either it is the right type of pollen or wrong type of the pollen. What is meaning of here right type of pollen? Compactability. Wrong type of the pollen is the incompatibility. See. So that's why it is the right type. Right type means compactability pollen. Compactability pollen. What about this one? Incompatibility. So that's why here this pistil, the pistil is a reject the which one? Incompatibility pollen grains. Which pollen grain is accepted? The compactability pollen grain. Why the pistil is recognized or accept the here the compactability pollen grain? Because see here the both contain the same genes. That's why here the pistil is accepting the compactability pollen grains. Here the genes are different. That's why what happened here? This kind of the pollen grains is rejected. These pollen grains cannot germinate on the stigmatic globe because the genes are different is there. That's why what happened here? Inhibit the germination of pollen grains on the stigmatic globe. Here the compactability pollen grains. When the compactability pollen grains is landing on the stigma, automatically here the pollen grains promote the further post-pollination events. That's why here it is promoted the post-pollination events. It is rejected from the here the stigmatic globe. The accepted the compactability pollen grains and rejected the incompactability pollen grains. See in this one, same S1 and S2, S3 and S4. In these crop plants, this is which kind of the pollen grains? This is known as the incompactability pollen grains. This one, compactability pollen grains. Incompactability, compactability. It is a same species pollen grain. They contain same genes S3 and S4. That's why this is the compactability on the stigmatic globe. It is a incompactability because this pollen grains is come from the tomato species. That's why it is on the stigmatic globe of the potato. This pollen grain is incompactable. This is compactable. This is known as here the pollen-pistil interaction. The pistil has ability to recognize either it is the right type of pollen or wrong type of the pollen. When the right type of the pollen, the stigmatic globe is accepted. When wrong type of pollen is landing on the stigmatic globe, they are rejected, and further the growth is prevented on the stigmatic globe. Suppose accepted pollen grains and further promoted the post-pollination events. See this one. Now, what is meant by here the pollen-pistil interaction? The pollen-pistil interaction means, so when, see here, all events from the pollen grains is landing or deposit on the stigmatic globe until the pollen tube is entered into the ovule, are together referred to as the pollen-pistil interaction. The all events from the pollen deposit on stigmatic globe until the pollen tube is entered into the ovule are together referred to as the pollen-pistil interaction. The pollen-pistil interaction is depending on the chemical components. The chemical components are released by the either it is the pollen grains or stigmatic globe. That's why definitely it is depending on the chemical components. And also the pollen-pistil interaction is one of the dynamic process. Why it is a dynamic process? Because it is further here accept the pollen grains and reject the pollen grains. Compatible pollen grains is accepting, incompatible pollen grains is rejecting. It is one of the here the main dynamic process. When the compatible pollen grains is there promoting, and when incompatible pollen grains is landing on the stigma, they are rejecting. This concept is known as here the pollen-pistil interaction. In recent years, the botanists have to be identified what are the chemicals is play role in the interaction between here the pollen and the pistil. What are the chemical components? He is accepting and rejecting the here the pollen grains on the stigmatic lobe. Even the incompatible pollen grains also used in plant breeding technique, and then that is also further used in the development of the hybrid varieties in plant breeding program. Okay, next one, how the percentage of the pollen germination calculate. How to calculate the percentage of pollen germination in laboratory? See here, in simple method is that first you take any flowers, balsam plant, vinca rosea, crotalaria, you take any one flower and the pollen dust is placed on glass slide. The pollen dust is placed on the middle portion of the glass slide, and then add the 10% of the sucrose solution. After adding 10% of sucrose solution, at least a minimum 15 to 30 minutes, 15 to 30 minutes, the pollen germination is takes place on glass slide. This glass slide is observed under the microscope. The clear visible, the pollen tube is comes out from the pollen grains. Okay, so how many pollen grains are germinated in particular area? Here one simple formula is that how to calculate the percentage of pollen germination. See, small n by capital N into 100. It is how to calculate the percentage of pollen germination. Small n by capital N into 100. What is here small n? The total number of pollen germinated. The total number of pollen germinated. What about capital N? The total number of pollen grains. For example, in this region, total number of pollen grains 50. In out of 50, at least for example, 25 are germinated. That's why small n, total number of pollen germinated, capital N, total number of pollen grains in particular area, microscopic field, into 100. That is 50%. It is the simple formula to calculate the percentage of pollen germination in laboratory also. Next, go for another topic. Artificial hybridization. artificial hybridization. In artificial hybridization, what is the artificial hybridization? So here the hybrids is developed by the human beings, plant breeder. Why here the hybrids are developed by the human beings? So here for development of the desirable characters in a single superior hybrid. That's why in this method, the artificial hybridization in this method, the combining of characters of species or varieties or even genera also, to obtain the desirable characters in a single superior hybrid. That is known as the artificial hybridization. Once again I repeat, artificial hybridization means it is a method of the combining characters of varieties or species or even genera to obtain the desirable characters in a superior single hybrid. So that is known as here the artificial hybridization. So first of all, in artificial hybridization, select the desirable characters of the crop plants. In desirable crop plants, first select the desirable crop plants. For example, this is a male plant, this is a female plant. See here. First, select the desirable characters of the parents, male parent and the female parent. They have very good qualities of the characters in their plants. First of all, in selected female parent, they contain bisexual flowers. So it is followed by the first step. That step is known as the emasculation. What is emasculation? Here see. Generate the bisexual flowers, that is referred to the self-pollination, because the pollen grains is easily landing on the stigmatic lobe. When the pollen grains is landing on the stigmatic lobe, automatically what happens here? The self-pollination takes place. That purpose here, suppose the selected female parent, they contain bisexual flowers. The removal of the anther from bisexual flower in bud stage. That process is known as the emasculation. Here the complete remove the anthers. The removal of anther from bisexual flower in bud stage is known as the emasculation technique. It is one of the technique in a plant breeding program. When the selected female parent, they contain the bisexual flowers, immediately the removal of anther. The removal of the anther. In bud stage, that is known as the emasculation. After the removal of the anther, that is followed by the second step, that is known as the bagging. You take any polythene bag or butter paper, that is covered here the emasculated flower. Emasculated flower means here anther or removed. This emasculated flower is covered by the one polythene bag or buttercup paper. That technique is known as the which one? Bagging. Third step, artificial pollination. What is artificial pollination? Artificial pollination. Already we have selected the desirable male parent. First of all, from this male parent, the collecting the pollen grains. After collecting these pollen grains and remove the polythene bag, and this pollen dust is placed on the stigmatic lobe. When this complete the pollen grains, the desirable pollen grains is collected from the parental plant, the male parental plant, and this pollen grains is dusting on the stigmatic lobe of the emasculated flower. That is, and again here rebagging. In artificial hybridization is followed by the main, these are 4 steps are there. One is emasculation, another one is the bagging, artificial pollination and rebagging. What is the significance of the here bagging? When the removal of the anther from the bisexual flower, that is immediately enclosed by the polythene bag, because it is prevented the contaminated unwanted pollen grains. In case another pollen grains is landing on the stigmatic lobe, okay, so that is either desirable characters or not desirable characters. That occurs here. What happened? The prevented the contaminated pollen grains, or it is prevented unwanted cross pollination. The prevented unwanted cross pollination with the help of the bagging technique. That is the main significance of the here bagging. It prevented unwanted cross pollination, or it is prevented the contaminated pollen grains. That is the main concept of the here bag. So finally here in artificial hybridization, it is followed by the 4 steps are there. One is the emasculation, second one is the bagging, and the third one artificial pollination, and the fourth one is rebagging. It is the concept of the artificial hybridization. Next one, fertilization. You know what about? Now we are going to discuss about the here fertilization. So what is the meaning of the here fertilization? Now fertilization. The fertilization means the fusion of male and female gametes. The fusion of male and female gametes is known as the fertilization. This fertilization process is involved by the 4 steps are there. What are the 4 steps that are involved in the fertilization process? First one is the germination of pollen grains. Germination of pollen grains. Second one, entry of pollen tube. Entry of pollen tube into ovule. Entry of pollen tube into ovule. Third one, the entry of pollen tube into embryo sac. The entry of pollen tube into embryo sac. Last one, fusion of gametes. These are 4 steps are involved in fertilization. What is meaning of here fertilization? The fusion of male and female gametes is known as the fertilization. The germination of pollen grains, the entry of pollen tube into the oval, the entry of pollen tube into the embryo sac, and the fusion of gametes. So first one, the germination of pollen grains. When the pollen grains are germinated, when the pollen grains is landing on the stigmatic lobe, the pollen grains is start the germination. For example, it is a stigma. First, the pollen grains is landing on the stigma. When the pollen grains is landing on the stigma, the pollen grains is receiving moisture from stigmatic lobe and sugar, and the pollen grains is get the swelling. When the pollen grains get the swelling, automatically what happens? The intake of pollen grains. This is the intake. The intake of the pollen grains comes out through the any one of the germ pore of the egg side to develop the pollen tube. It is a pollen tube. It is the outermost layer of the pollen grain. It is a germ pore. This is known as the intake. Already know about intake, the inner wall of the pollen grain. Outer wall of the pollen grain is the exine. The inner wall of the pollen grain comes out through any one of the germ pore of the exine to form the pollen tube. The pollen tube is down. This is. When the pollen tube comes from the pollen grain, that is indicated the germination. This is. The pollen grains are germinated on the stigma. Many compactable pollen grains are landing on the stigmatic lobe. But only one pollen tube is finally entered into the ovary. It is very important. Many compactable pollen grains is landing on the stigmatic lobe, but only one pollen grain, the pollen tube, is entered into the structure of the ovary. See here, in this one, it is pollen tube. This pollen tube is contained the vegetative nucleus. It is a vegetative nucleus. Vegetative cell nucleus, and here the generative cell is undergo the meiotic cell division to form the two male gametes. Some scientists is strong belief that the vegetative cell of the nucleus is always placed in the terminal part of the pollen tube. Some scientists is believing that the vegetative cell nucleus is only initially is located in the terminal part of the pollen tube. Different opinions are there. See here, the two main gametes are developed from the generative cell. This is the vegetative cell nucleus that is located in the terminal part of the pollen tube. Okay. So what are the elements are essential for the germination of the pollen grains? It is very important. One is the boron, another one is the calcium. These two elements are required for the germination of the pollen tube. Boron and calcium. The more amount is boron. A little bit calcium is also required for the germination of the pollen grains. What is optimum temperature for the germination of the pollen grains? The optimum temperature 20 to 30 degrees. 20 to 30 degrees, it is a optimum temperature for the germination of the here pollen grains. That is. These are essential. And one more important point, the two male gametes both are non-motile. There is no motility. That's why here the pollen grains is carried by the pollen tube. Sorry, the male gametes is carried by the pollen tube. Total one pollen tube is developed from the one germ form. This condition is known as the monocyphonous. That condition is known as the monocyphonous. The monocyphonous means the pollen grain is contain only one pollen tube. It is the most of the angiosperms. Only one pollen tube is developed from the each pollen grain. Example, the most of angiosperms. One of the best example, capsule. Next one, polycyphonous. What is the meaning of here? Poly means many. The many pollen tubes. Many pollen tubes. Many means maximum 3 because the 3 germ pods are present in dipods. So generally one germ pod is present in monopods. Monopulpate and the poly or tripulpate. In polycyphonous means more than one pollen tube is developed from the pollen grains. That is known as the polycyphonous. So it is reported in Malvaceae family and Cucurbitaceae family. Malvaceae family and the Cucurbitaceae family, in these two families they can found polycyphonous condition. Okay, the most of the angiosperms, that is the monocyphonous condition. What is meant by monocyphonous? The one pollen tube is developed from each pollen grain. What is the polycyphonous? More than one pollen tube is developed from the pollen grain. That is known as the polycyphonous. Okay, next one. This pollen tube was discovered by the G.B. Emasculation in Portulaca plant. The pollen tube was discovered by G.B. Emasculation in Portulaca plant. Next one, the longest pollen tube is reported in Geomais. The Geomais is contained the longest pollen tube. Longest pollen tube. So this is the information about the first step of the fertilization, that is the germination of pollen grains. Second step, the entry of pollen tube into the ovary. See here the pollen grains so germinated on the stigma. This pollen tube, one more important point is that here, this pollen tube is a release of some enzymes. The best example, pectinase enzyme. This enzyme is responsible for the disintegrate the tissue of the stigma and the style. The stigma and style tissue is damaged and create the one pathway for entry of the pollen tube inside of the ovary. That's why here the pollen tube is also release of the some of the chemicals or enzymes. These enzymes are responsible for the pollen tube is easily enter into the ovary. Okay, the stem is a solid tissue. This solid tissue is broken by the pectinase enzyme. Okay, so next one, the entry of the pollen tube into the ovary. So finally here, see this is the structure of the ovary. In central position, embryo sac, nucellus, integument, outer integument and inner integument. See this. It is one of the anatropous ovary. See in this anatropous ovary, this is a pollen tube. Gradually the pollen tube comes down and enter into the ovary. This is known as the microphyte. It is very important here. Already we have discussed about the main one of the special structures from the funicle, that is known as the obturatus. The obturatus is developed from funicle. These are the filament-like structures. These filament-like structures are responsible for the guiding of the pollen tube is entered into the ovule. This point is very, very important. This operator is responsible for the guiding of the pollen tube is entered into the ovule only. That's why here see the pollen tube is entered into the ovule. It is followed by the total 3 methods are there. One is the pollen tube is entered into the ovule through the microphyte. This is male gametes. This is the vegetative nucleus. See here in this method. So that is the first one, phorogamy. Second one, chalasogamy. Third one, mesogamy. The entry of pollen tube into the ovule is followed by the 3 ways are there. That is the 3 pathways are there. That is known as the phorogamy, chalasogamy, and the mesogamy. What is meant by here phorogamy? Very easy definition. The phorogamy means the pollen tube is entered into ovule. The pollen tube is entered into ovule through the microphyte is known as the phorogamy. That is known as the phorogamy. The most of the angiosperms, the pollen tube is entered into the ovule through the microphyte. The best example, polypetale. What is the polypetale? It is a subclass of the dicots. The most of the polypetale members here, that is the phorogamy takes place. Next one, what is chalasogamy? This is a phorogamy. What is meaning of here the chalasogamy? The chalasogamy means here the pollen tube is entered into the ovule through the chalasa. See. The pollen tube is entered into the ovule through the chalasa. That is known as the chalasogamy. chalazochemy. It is found in Casuarina. This chalazochemy is first reported by fruit scientists. Next one, mesogamy. See here. This is the mesogamy. Pollen tube. The pollen tube is entered into the oven through the integuments. Through the integuments, that is known as the mesogamy. The best example for the mesogamy, cucurbita. These are three pathways the entry of pollen tube into the oven. What are the names of the three pathways? One is forogamy, chalazochemy and mesogamy. What is forogamy? The pollen tube is entered into the oven through the micropyle. What is chalazochemy? The pollen tube is entered into the oven through the chalaza. What is mesogamy? Here the pollen tube is entered into the oven through the integuments or funicle. That is known as the mesogamy. These are the examples. Okay, so this is the second step, the entry of the pollen tube into the oven. Next, go for the third one, the entry of the pollen tube into the embryo sac. So next one, the entry of the pollen tube into the embryo sac. Already know the structure of the embryo sac. The total 8 nucleated and 7 cells. It is an egg apparatus towards the microphylla region. These are 3 cells towards the chalazal region. In central position, polar nuclei. Okay, it is an egg cell. Or it is also known as the male gamete. Male gamete. So now in this, on either side of the egg cell, these are the synergid cell. You know already here the synergid cells contain a finger-like projections. These are finger-like projections is known as the filiform apparatus. The filiform apparatus. So here the pollen tube is entered into the oven through the ending of the method, that is mostly forogamy. Here this is a pollen tube. The pollen tube is carried by the 2 non-motile male gametes, and it is a vegetative cell nucleus. Vegetative cell nucleus. See. This pollen tube is entered through the micropyle. Towards the micropyle region, the egg apparatus is there. This is the egg apparatus. Why particularly here the pollen tube is attracted towards the micropyle region? Because the presence of the here the filiform apparatus. What is the main function of here the filiform apparatus? 2 is there. One is the supplying of the food material from the nucellus to the development of the embryo sac. Second one, the filiform apparatus is the release of the some chemicals. These chemicals is responsible for the the guiding of the pollen tube is entered into the embryo sac. It is very important. So which one is responsible for here the entry of pollen tube into the oval apparatus? Apparatus is responsible for the entry of the pollen tube into the oval. So which one is responsible for here the entry of the pollen tube into the embryo sac? The filiform apparatus. Apparatus that is responsible for pollen tube is entered into the oval. The filiform apparatus is responsible for the pollen tube is entered into the embryo sac, the structure of the embryo sac. See here. Okay. Now the pollen tube is entered into the micropyle towards the evaporator region. Now, with the help of the chemicals, the pollen tube is attracted towards the microphylla region. That's why here the pollen tube is attracted to the microphylla region with help of the chemicals. This is known as the chemotrophic movement. The pollen tube growth is known as the chemotrophic movement. The growth is controlled by the chemicals. That's why the pollen tube growth is known as the chemotrophic movement. And finally here the pollen tube is entered into the one of the synergid cell and destroying of the here one of the synergid cell, and then this pollen tube is the gradual growth inside of the embryo sac. See here. That's why one of the synergid cell is degenerated, and when the pollen tube is entered into the structure of the embryo sac, the pollen before here the pollen tube is entered into the structure of the embryo sac, the vegetative cell nucleus is degenerated. Here already one synergid cell is degenerated. And later here the pollen tip are broken and the release of the two male gametes. The two male gametes are released in the structure of the embryo sac. Okay. Next one. Here, the particular in this region, two X bodies are there. What are the X bodies? The X bodies means these are a dark color granules are reported in the structure of the embryo sac. One of the X bodies developed from here the degenerated one of the synergid cell. Another one is developed from the degenerated the vegetative cell nucleus. These are two X bodies are reported in the structure of the embryo sac when the pollen tube is entered into the structure of the embryo sac. Okay, this is the third step, the entry of the pollen tube into the embryo sac. And fourth step, the fusion of gametes. Fusion of gametes. Here total how many male gametes are there? The two male gametes are released inside of the embryo sac. Total two male gametes are released in structure of the embryo sac. Okay, so the fusion of gametes, XL or female gamete. The XL is present in the egg apparatus. Is one of the largest cell in egg apparatus. It is located in central portion of the egg apparatus. On either side, synergid cells are there. Now the fusion of the gametes. First fusion. See here. In two male gametes, one of the male gametes are fusion with the egg cell. Male gamete fusion with the egg cell nucleus. Or female gamete. Female gamete. That is male gamete plus XL nucleus or female gamete. Now, the fusion of male and female gametes, is known as the syngamy. It is the true fertilization. The fusion of male and female gametes is known as the syngamy. As a result of the syngamy, a diploid zygote is developed. 2A. What about male gamete nucleus? Haploid. The female gamete nucleus? Haploid. When the fusion of two haploid nucleus, that is developed the diploid condition of the zygote. So this is known as the syngamy. What is the meaning of syngamy? The fusion of male and female gametes is known as the syngamy. Second one, another male gamete. The male gamete is a fusion with the secondary nucleus. What is secondary nucleus? The before or after entry of the polytube, the two polar nuclei, this is the two polar nuclei fuse together and form the secondary nucleus. This secondary nucleus is contained total two nucleus. This is. That's why another male gamete is a fusion with the secondary nucleus. Second male gamete or fusion with the secondary nucleus. It is a haploid. It is a diploid. So when the second male gamete is a fusion with the secondary nucleus, it is known as the triple fusion. Why it is called triple fusion? Three nucleus fusion takes place. So what is a triple fusion? The male gamete is fusion with the secondary nucleus is known as the triple fusion. As a result of the triple fusion, that is developed the pen. What is the full form of the pen? The primary endosperm nucleus. The primary endosperm nucleus. It is a triploid condition, 3N triploid condition. So this is the first one. The male gamete or fusion with the female gamete is known as the syngamy. As a result of syngamy, a diploid zygote is developed. Second one, the male gamete is fusion with the secondary nucleus is known as the triple fusion. As a result of triple fusion, pen is developed. That is primary endosperm nucleus. The primary endosperm nucleus is the triploid, zygote is the diploid. And further, this zygote is developed into the embryo. What is the main function of primary endosperm nucleus? This primary endosperm nucleus is further developed into the endosperm from the central cell. It is a nutrient tissue. The food material is supplied to the development of the embryo. Okay. At that time, in the structure of the embryo sac, the two fusions takes place. One is the syngamy, another one is the triple fusion. The syngamy is the true fertilization and the triple fusion is the vegetative fertilization. Okay, it is a true fertilization, it is a vegetative fertilization. First here, the syngamy was discovered by the Strasburger in Monotropa. The syngamy was first discovered by the Strasburger in Monotropa plant. The triple fusion is discovered by the Navasin in Lilina or fritillaria. At the same time, the two fusions are takes place in the structure of the embryo sac. It is known as the double fertilization. It is very important. It is unique characters of the angiosperm. In angiosperms only the double fertilization takes place. Total two fertilizations are takes place. One is the syngamy, another one is the triple fusion. At a time, the two fusions are takes place in the structure of the embryo sac. That's why that is known as the here double fertilization. Even the double fertilization was discovered by the same scientist Navasin in Lilium and Fritillaria. Okay, this is the concept of the fusion of gametes. Here total how many gametes are fusion? Here total 3 gametes are there. 2 male gametes and 1 female gamete. How many nucleus fusion takes place here? Total 5 is there. One is the male gametes 1 nucleus, another male gamete 1 nucleus, female gamete 1 nucleus, and secondary nucleus 5 nucleus. It is very important in double fertilization. The total 3 gametes are involved and 5 nucleus fusion is takes place. That is the characters of the here double fertilization. Here one more important point: the male gametes is generally here the non-motile. This non-motile male gametes is carrying through the pollen tube. That's why this is known as the siphonogamy. The non-motile male gametes is carried by the pollen tube is known as the siphonogamy. It is the characterization of the here the fertilization. Okay, so.
Subscribe now and press the bell icon. Never miss an update.